Welcome to Exam Mini Video 1. Today we are talking about X inactivation and X-linked inheritance. To begin, here are some review terms. Please pause this video and study them if anything seems unfamiliar. Here are some new terms for your reference. Since this video topic concerns the X chromosome, we are talking about sex chromosomes. Female cells contain 2 X chromosomes, while male cells contain 1 X chromosome and 1 Y chromosome, each of which code for different protein synthesis, which causes a problem. Female cells are synthesizing twice the amount of X-coded protein as male cells. Enter the Lion hypothesis, which states that in order to equalize the amount of produced protein, the inactivation of an X chromosome occurs randomly in somatic cells, meaning body cells, at some point in development. This shuts down the protein synthesis from one X chromosome and equalizes the amount of X chromosome proteins being produced between females and males. This inactivated X chromosome is the Barr body. Let's see this in practice. For the purposes of this demonstration, a turned-on light bulb represents activation, and a turned-off light bulb represents inactivation. The gene that codes for the color of a cat's fur is found on the X chromosome. The capital B is the dominant allele that codes for black, and the lowercase b is the recessive allele that codes for yellow. If we have a cat who is homozygous dominant, it does not matter which X chromosome is inactivated. Either way, an X chromosome with a dominant allele is active, so every cell expresses a black fur phenotype, and we get a black cat. The same applies to a cat who is homozygous recessive, except that it expresses the yellow phenotype. But what happens if we have a heterozygous cat? In this case, X inactivation does matter. Because X inactivation occurs randomly, some cells, like the one on the left, will have the dominant allele activated, producing the black phenotype, while others, like the one on the right, will have the recessive allele activated, producing the yellow phenotype. We then get a mixture of black and yellow fur via the randomized expression of the alleles, giving us a tortoise phenotype cat. Let's look at another example where we consider an additional trait in cats that is autosomal, hair length, for which long hair capital H is dominant to short hair lowercase h. What happens if we cross a heterozygous long-haired black male cat with a short-haired yellow female cat? As we can see in the Punnett square for coat length, half of the progeny will be heterozygous long hair and the other half will be short hair. In the Punnett square for coat color, we see both X-linked inheritance and X inactivation at work. Half of the progeny will be heterozygous females because all the females receive a dominant allele from the father and a recessive allele from the mother. The other half of the progeny will be yellow males because the male progeny inherit the Y chromosome from the father, which has no X-linked genes, and the only X chromosome they can receive from the mother has the recessive allele. Let's complete a branch diagram to find the probability that an offspring could have any combination of the phenotypes. We must list the possible progeny phenotypes for coat length and their probabilities, and then multiply those probabilities by those of the coat color phenotypes. As we can see, a quarter of the progeny will be long-haired tortoise females, a quarter will be long-haired yellow males, another quarter will be short-haired tortoise females, and the final quarter will be short-haired yellow males. Since all the female progeny will be heterozygous for coat color expressing the tortoise phenotype, none of them will be visually the same because of the randomized pattern of X inactivation. That concludes this mini video. Stay tuned for another in 2 weeks.